While on one of his late-night strolls, a crippling feeling of fear befalls John. He feels like he's being watched, like something sinister is lurking in the shadows and is out to get him. He feels the sudden urge to run. This feeling has been reoccurring for John, and to understand its root cause, he decides to consult a psychiatrist who eventually attributes his paranoid behavior as being a symptom of schizophrenia. As an epidemiologist, John knows that this diagnosis will have life-changing consequences. He knows all about the social isolation and the stigma associated with this condition, as well as the more practical hurdles, such as the very high unemployment rate of between 70 and 90% and the very high suicide rate of around 5% amongst afflicted patients. Roughly 1 in 100 people will develop schizophrenia in their lifetime, and while it generally first appears in adolescence, occurrences of the disorder in childhood and late adulthood have been documented. It is important to note that schizophrenia tends to affect more men than it does women. Schizophrenia presents as a constellation of symptoms that can be segregated into 3 main groupings: reality distortions, such as delusions and hallucinations, disorganizations in terms of cognition and behavior, as well as negative symptoms such as the flat effect, poverty of speech, reduced activity, and catatonia. Early treatment may help get symptoms under control before serious complications develop, but treatment will likely have to continue throughout the individual's lifetime. Although in popular media, hallucinations are oftentimes synonymous with schizophrenia, of the people with schizophrenia, only around 27% will report visual hallucinations, and only 59% will report auditory hallucinations. One of the biggest success stories in contemporary schizophrenia research has been the confirmation of the long-held suspicion that the causes of the disorder involve events in early life, at birth, or even in utero. The view emerging from this work is that schizophrenia is fundamentally a disorder of disrupted neural connectivity. The neurodevelopmental theory of schizophrenia has been established based on the finding that early developmental brain abnormalities interact with the normal maturation process to culminate in the onset of schizophrenia. These abnormalities, often but not exclusively found in the frontal cortex, present in the form of mild IQ disadvantages, delays in reaching developmental milestones, as well as speech and language problems, and are all associated with an increased risk of developing schizophrenia. There is a strong influence of genetic factors in determining risk for schizophrenia, with a spectrum of risk alleles, common and rare, with each allele contributing only a small fraction to the total variance. Schizophrenia tends to run in families, but no single gene has been shown to be responsible. It seems to be the case that different combinations of genes make people more vulnerable to the condition. Likewise, environmental factors also contribute to the development of schizophrenia, such as obstetric complications, infections, winter or spring births, migration, urban living, childhood adversity, and cannabis use. When put together, however, it is important to appreciate that certain environmental exposures are only of consequence given a specific genetic makeup. This is to say that certain genes moderate the effects of certain environments and vice versa, meaning that both the genetic and environmental causes of schizophrenia must be recognized together when trying to explain its etiology. While the relative importance of genetic and environmental factors is debated, what remains clear is that the phenotype of schizophrenia begins as neurons fail to form and migrate in the early developmental stages, long before any behavioral phenotype is obvious. Defective neurogenesis has been reported in animal models and patients of schizophrenia alike. More specifically, in across models, we see consistent reductions in neural stem cell proliferation, decreased cerebral and hippocampal volume, and increased ventricular volume and defective neuronal migration, amongst other defects. These findings are consistent with the large number of genes that are associated with schizophrenia, as well as their involvement in different aspects of neurogenesis. One such example is the DISC1 gene, standing for disrupted in schizophrenia, which has been shown to regulate neural stem cell proliferation by regulating AKT glycogen synthase kinase 3 beta signaling. When DISC1 expression is disrupted, as it is in schizophrenia, neuronal maturation is significantly altered depending on tissue-specific and age-specific factors, meaning that there is a clear evidence of a causative link between defective neurogenesis in utero and eventually developing schizophrenia. At this point, We have already described a few of the etiological theories of schizophrenia. It would now be interesting to discuss a recent paper published in 2022 exploring the neurodevelopmental hypothesis of schizophrenia, which induced stem cells to test for migration. The neurodevelopment theory of schizophrenia was first described by Dr. Daniel Weinberger in the 1980s. This theory coincided with some independent initial work which led to the discovery of stem cells. At the time, no connection between the mental illness and the exciting new cell type was proposed. Throughout time, there was increasing evidence of genetic basis of schizophrenia. Today, we consider the heritability of schizophrenia to be 0.81. Thus, 81% of the variability observed in schizophrenia seems to be attributable to genetic factors that underlie and influence everything from cellular mechanisms to human behavior and physiology. A major advancement in stem cell biology occurred in the early 2010s, as a method was developed to induce pluripotent stem cells from somatic terminally differentiated cells. Protocols were then developed to differentiate these cells into nearly any cell type that scientists needed to use in research. In the field of schizophrenia, this represented a chance to test for the neurodevelopmental hypothesis, whereby allowing for the reprogramming of cells taken from schizophrenia patients into neural stem cells for analysis. Direct sampling of human neural stem cells is difficult to do during life, as the cells are housed deep within the brain's subgranular and subventricular zones. The only viable method of extraction would be an autopsy. Autopsies produced histological evidence of impaired cell migration, but the viability of those cells for expansion and culture was poor. Yamanaka factors, which are center transcription factors such as OCT4, KLF4, CMYC, and SOX2, must be introduced exogenously to promote reprogramming and pluripotency. These factors are naturally highly expressed in embryonic stem cells. Recently, mounting evidence has shown that the introduction of these factors leads to mutations and higher levels of heterogeneity in induced stem cell populations. These genetic changes are a serious concern when studying potential differences in gene expression and activity that may be causing impaired neuronal migration as seen in schizophrenia. Overall, as the previous data might be questionable, Lee and colleagues sought to use a novel new molecule inhibitor-based lineage switch method developed in 2017. This protocol allowed for the induction of neural stem cell-like cells from human adipose-derived stem cells with little genetic consequences. The group used cells derived from 9 patients, which are divided into 3 groups. The first group contained 3 people with a schizophrenia diagnosis, according to the definition within this Diagnostic and Statistical Manual for text revision. The second group contained 3 people who are at high genetic risk of schizophrenia, defined by at least 2 relatives with schizophrenia, one of which must be first degree or with a monozygotic twin who had been diagnosed. The third group had 3 healthy control participants, free from any history of psychosis and all psychotic disorders. Cells were harvested via biopsies, induced into neural stem-like cells, and then assayed. The assumption was made that little genetic aberrations were produced by the induction protocol. Neurospheres were formed to assess the stem cell nature of the resulting cells. Expression levels of specific markers of neural stem cells were also assessed by quantitative PCR controlled by GAPDH. Interestingly, this assay yielded a reduction in SOX2 that was not statistically significant. This result would have been masked by a standard reprogramming protocol with Yamanaka factors. Moreover, this supports recent literature suggesting that SOX2 is decreased in psychosis and increased after treatment with antipsychotics. This result highlights the importance of a varied array of methods and assays, as the preparation of samples can possibly affect notable results. Despite this finding, the researchers didn't attribute the decrease to any meaningful pathological process. The central aim of the paper was to assess the capacity of the neural stem cells to migrate, as impaired cellular migration contributes to the inability of newly birthed neurons to functionally integrate into parts of the brain where they are needed. The control and genetic high-risk groups had migration data that fit an exponential decay function. Within the first 24 hours, the cells migrated over 80% of the distance they would travel in 48 hours. The cells from schizophrenic participants traveled at a more consistent rate. with data fitting a quadratic function. Notably, a high-risk individual was later diagnosed with schizophrenia. This participant also showed an intermediate phenotype between health and schizophrenia migration data, further suggesting that cell migration is affected in schizophrenia. The conclusion from this recent study using stem cells in schizophrenia is that SOX2 gene expression is reduced, which may impact neurogenesis, a process that has been shown to be involved in the development of schizophrenia. Impaired migration may be permissive of schizophrenia. Despite this, the study used a small homogeneous sample, which should be addressed by further studies. The sample used does not reflect the sex ratio in schizophrenia diagnosis statistics. Furthermore, the sample molecule inhibitor lineage switch protocol employed isn't widely utilized. Thus, there may be problems in replication, or in the validity of the method may not be fully independently verified to produce less genetic modifications than the widely used Yamanaka factors in somatic reprogramming. To continue basic biological research in schizophrenia neurobiology, we propose an experiment to examine schizophrenia patient-derived stem cells in cerebral organoid model systems. The organoids would be formed from healthy participant stem cells, allowing for the modeling of a healthy nervous system. Following organoid establishment, participant-derived cells would be reprogrammed using classical methods and modified for lineage tracing with fluorescence proteins under the transcriptional control of a neuron-specific promoter. Cells would then be transplanted into the organoids and monitored, as the goal would be to further characterize the neurodevelopmental aspects of schizophrenia. Migration of the cell will be measured. Furthermore, cell proliferation and the orientation of division will be assessed, as these factors could assist in the pathophysiology of impaired migration. This design will add artificial heterogeneity to cellular genetics by using a sample from a healthy person for the organoid, then studying the cells that are transplanted, which would be derived from one of the experimental groups. However, the intention of the study is to characterize cells from schizophrenia diagnosed people in a model system with more complexity than an in vitro 2D culture, but that is not as complex as the animal models where schizophrenia is often induced by physical or pharmacological methods that do not well represent the genetic underlay that seems important in human psychosis. This experiment, as a proof of concept, could be then expanded to produce cerebral organoids purely from a single participant to recover the genetic identicality that would be seen in human cases of the disorder. A similar design would be taken for participant selection as the study that was previously discussed, where 3 groups would be used: a schizophrenia diagnosis group, a group with high genetic risk, and a healthy control group. Importantly, we will pay special attention to equity, diversity, and inclusion to ensure that the proper sex and ethnic ratios of our sample is seen in the diagnostic data, and to attempt to represent the vast heterogeneity of schizophrenia diagnosis. Works of research like the ones discussed in this presentation are aimed at helping patients like John live relatively normal lives in spite of their diagnoses.
Created using Powtoon.